So you can go into Canvas and see what your current grade is. Okay. And you can also notice that I put in a new assignment, which is your extra credit assignments. And this extra credit assignment is something that you are going to submit online, and it's optional, and it doesn't count into your grade. And it is due the Monday of finals week. And so if you click on this, what you'll notice is there's a short description of what it is that you're writing about. So what I would like you to do is to type up some information about something that we talked about in class, but it doesn't, I don't want it to be just like what we went over, like you just copy down your notes. Some people do that. But what I'd rather have you do is, like, if you went out and found an article, like, say, for example, on a genetic disease, or on, we're going to talk about human chimeras, for example, today. Then what you're going to do is you're going to take that information and then you're going to summarize it and you're going to include the references. So this is going to be kind of analyzed by a program which most of you are probably familiar with, which is called Turnitin. And so it'll tell me if you have taken the information verbatim. So like if you just copied and pasted it, then it would tell me that you did that. And it would give you a red color when you uplink it. So what you want to do is you want to make sure that you don't have a lot of copying of the information, but you need to put it in your own words, okay? So this is one way, if you didn't do so well on the midterm exam, to help improve your grade. It's also a way to make up for if you missed a lab. Okay, so if you've already missed a lab, you'll definitely want to do this. If you didn't do so well in the midterm, you definitely want to do this. What I found is that people that kind of need it the most are the ones that don't do the extra credit, and the ones that are, a solid B or a solid A are the ones that do the extra credit. So you want to make sure that you take advantage of that if that is what you so desire to do. Okay. Well, also, I haven't had the time to put it into the Canvas, but you'll also have another assignment, which is going to be on cell division, which is going to be due on Sunday at midnight. We're not going to have a quiz this week. So our first quiz after the midterm is going to be next Monday. Okay. So we'll have a quiz next Monday over the information provided this week, an in-class quiz. We're also having lab today, so make sure that you come to lab. Are there any questions about anything? No? Okay. So we were talking about meiosis as a special type of cell division that produces haploid cells. And remember that this only takes place in the ovaries and the testes. And the reason for this is to create cells that when they then combine can produce offspring that are genetically different from the parents. So one thing about meiosis that's really interesting is that it also involves what I try to call genetic shuffling of the information between the chromosomes. So remembering that meiosis is important in producing offspring that are genetically different than the parent, okay? So this is sexual reproduction, and it involves genetic recombination, right? When the egg and the sperm come together, to produce a new individual. Okay, so there is a mechanism that is really interesting because you would think that would be very detrimental, but it actually, the benefits of it seem to outweigh the costs. And this process is what is referred to as crossing over. So crossing over occurs in prophase 1. So remember that meiosis has one cell division and then the second cell division. So this would be at the very beginning of meiosis in prophase 1. And so this involves homologous replicated chromosomes coming together. So the chromosomes are still replicated. They come together and they swap genetic information. So I like to think of it as shuffling of card decks. Right? So if you imagine one set of chromosomes being red and the other being black, and so you have a red and a black deck of cards, when they come together, they essentially shuffle, and the genetic material gets crossed over. And so what this means is that you have more genetic diversity than if you didn't cross the chromosomes over, okay? So let me just give you an example of this. So let's say I have one replicated chromosome, and I have a set of genes on this replicated chromosome that are A and B. And we'll talk about what those capital letters mean today. And then I have another set that has a different combination, different forms of the gene, are little a and little b. Okay. And so notice that if these behaved without crossing over, if they separated out, then capital A and capital B would always be bound together. So there's no way that a person would inherit a chromosome with a big A and a little b, or a little a and a big B, okay? So what happens during crossing over that is so important is that we have the exchange of information, right? So that in this next set, for example, we could have something that looks like this. So we would have big A, little b, right? Big A, big B. In this one, we would have little a, big B, little a, little b. Okay. And so what this does is it allows for more genetic variation because the genes can move around between chromosomes. Now the problem here is that this could lead to bad things happening like mutations. So the cost would be mutations. Mutations are neither bad nor good, right? It just has whether or not they have an effect on the offspring. And so, for example, if a gene crosses over in the middle of itself, then you can have a bad mutation that wouldn't code for a necessary protein, and then the egg or the sperm could be inviable, the embryo could be inviable, or a person could be born with a mutation. So the cost is that you could possibly get mutations. But the benefit seems to be, or must be, increased genetic variation. And the reason why this increase in genetic variation is so important is it allows populations to evolve. So for example, if everybody is susceptible to a particular virus, and we all die out because of one particular viral strain, then that would be bad from a bad perspective from a population genetic standpoint. But if we have genetic variation and some individuals are able to withstand the virus where some individuals aren't, then those individuals that are able to have that genetic variation are able to pass their genes on to the next generation. And so this idea of genetic variation is important at a population level. We'll also kind of talk about how it seems to be important as an individual level as well. Okay? But that increase in genetic variation is what makes crossing over so significant. So I just drawn here the exchange of one gene, B, right? But really it's a whole shuffling down the entire length of the chromosomes. And so in your book, they designate this with red and blue, okay? So they use A, B, and C. But notice here, this is crossing over, okay? And so when you see chromosomes, when you see chromosomes that have a combination of red and blue arms, then you know that crossing over had to have occurred, okay? So when you see this, that is crossing over, okay? So if we look at the diagrams that show meiosis, that one doesn't show the crossing over. Let's see if we can find one that does. Here's the crossing over. So notice that the chromosomes cross over and then they separate during meiosis 1, and then they separate again during meiosis 2. And so you eventually have, right, cells that have only half of the genetic information, right? So you have these haploid daughter cells, and the chromosomes are different than the chromosomes from the parents, from one individual parent. Okay. So this just shows the ending results of meiosis 2. Are there any questions about that idea? Oops. Okay, so we're going to talk about it in a little bit more detail, meiosis. So these are 2 examples of meiosis. This is called gametogenesis. So if you think about what genesis means the production of, right? Gameto means the production of gametes. So we have spermatogenesis. Right? This takes place in the testes. It is meiosis that produces them. And for one diploid cell, we produce 4 haploid cells. Right? The haploid cells are the sperm, and the sperm are very small in general. Like if we looked at different species of animals besides us, when you're trying to determine what is, if you look at a species you never saw before, and you're trying to determine which ones were male and which ones were female, you would actually look to see which one produces the smaller gamete, and that by biological definition is the one that is male, okay? So they are very small, and so they actually shed the extra cytoplasm. So if we look at what a mature sperm looks like, it has the nucleus filling up the majority of the head. There's just a little bit of cytoplasm. Then we have a mid piece, which has special types of mitochondria in it. And then we have part of the cytoskeleton, which is called the flagellum. So this would be the head. This is my nucleus. This is the mitochondria. And this is the flagellum. So that is protein, part of the cytoskeleton. And so it's kind of interesting because the mitochondria use fructose that is secreted by glands that the male has in his reproductive system. So they use the sugar to produce energy to power the flagellum so that they are not only small, but they're mobile, right? So they actually, the sperm actually have to swim up through the vagina, up through the cervix, up through the uterus, get into the fallopian tubes, and that is where fertilization takes place, right? And they're all in competition with one another, right, in order to kind of get to the egg first, because they're the ones that would be able to pass their genetic material on to the next generation. Okay. So if we compare this to oogenesis, which is the formation of the egg, right? Oops, genesis, I spelled that wrong. Okay, so this is the formation of the egg. So this takes place in the ovaries. Right? This is also meiosis. Right? But however, we have one diploid cell giving rise to one haploid cell. So the question is, where did all that extra genetic material go? And why is it seems to be inefficient? If we can produce 4 haploid cells, 4 eggs for every cell that's undergoing meiosis, why do we only produce 1? Does anybody know the answer to that? Why do we just produce 1 egg for every diploid cell that goes through this cycle? Well, we have multiple first. Well, only for one cell that's undergoing mitosis, you only get 1 haploid cell. Yeah? You know what it is?
Is it because when sperm are released, there's multiples going to make?
That could be a good explanation, but the most important thing is that it's got to be large. And so one nuclei gets all the cytoplasm, and the other nuclei are just kind of left out of the show, right? So this is what is called unequal cytokinesis. So we talked about cytokinesis as being the separation of the cytoplasm, and this unequal means that one nucleus gets all the cytoplasm. Because we need large eggs, because if you think about it, you know, for the first week, that's the nutrition of the embryo. And the egg has to divide and divide and divide and divide and produce a multicellular embryo before it actually implants in the uterus. So it needs lots of cytoplasm to start out with. Okay, so that's why the egg is big, because it is providing the nutrition for early embryonic development. So if I'll show you an image of this. So this is my diploid cell with the nucleus, and it divides, and one nucleus doesn't get any cytoplasm, and the other cell gets it all. Okay. This, so this is diploid. That would be meiosis 1. This is called a polar body. And it actually kind of hooks on to the cell, so it actually would be kind of like sitting there, right? So this is the polar body. That's why it's called polar, because it's kind of like on the edge of the cell, right? But this would be the polar body. And then when this cell undergoes cell division again, only one nuclei gets all the cytoplasm, right? Actually, I drew that too big, right? Okay. And so this is the egg. So the egg is what is released during ovulation, right? And it's picked up by the fallopian tube or the oviduct. And then the egg and the sperm come together to form the zygote. Okay. So this being unequal cytokinesis, the sperm, right, is equal cytokinesis. So spermatogenesis would be equal cytokinesis, because each nuclei, each set of chromosomes gets the same amount of cytoplasm. But then in the end, they shed it, right, because they got to be fast. So if they were too slow, then they wouldn't be the ones that get to the egg in time to fertilize it. Okay. So in your book, they have images. So you do not need to know the names of these. So they have like spermatogonium is the diploid cell, and then after meiosis 1, it's called the secondary spermatocyte or spermatocyte. You don't need to know the names, okay? So the sperm are just very small, have flagella, and have specialized mitochondria that power them, okay? So the sperm are stored in the epididymis, and then upon ejaculation, they move up into the body cavity, picking up nutrients and other things before they are ejaculated. If you compare this to oogenesis, and again, you don't need to know, you don't need to know the specific names. Yep, question?
Yeah, no question. So how does the process work then? Why are some sperm are like male and some are female, right? I mean, they're genetically.
We'll talk about that. Yes.
I'll wait.
Yeah, okay. So here, you don't need to know the name, right? This is the polar body, right? And then don't worry about the fact, one of the really kind of interesting things that you learn in AMP is that meiosis doesn't actually complete until the sperm fertilizes the eggs. So meiosis is kind of, the egg is arrested in meiosis 2, metaphase and meiosis 2, and then upon fertilization, then it completes it, and then you get that subsequent meiosis 2 completing, and then you get the zygote.
So the two polar bodies where the nucleus is split into two.
They're not, the nucleus doesn't, well, yeah, the nucleus splits in 2. So here, for example, one of the cells got all of the cytoplasm, and then this would just be the nucleus. And so it would also have a nucleus in there. Yeah. So it's just a way to get rid of genetic material, and it seems really wasteful, but if you think about it, we need to produce large eggs, and so that's just the mechanism by which we do it. Oops. Boy, this is touchy today. Okay. So fertilization. So when the egg and the sperm come together, right, they produce the zygote. They come together and they produce the zygote. So remember that the egg and the sperm are haploid cells, and the zygote is a diploid cell. So it's kind of interesting, the whole mechanism of fertilization. The sperm actually digests its way through this layer of cells that surrounds the egg. And so here's a picture of it. Here's the sperm, obviously much smaller than the egg. And it releases enzymes, and then it gets in there, and then it fuses, and then the two nuclei, the nuclei from the egg and the nuclei from the sperm come together, and then that becomes the diploid cell. Remember that I mentioned that the mitochondria of the sperm do not make it into the inside of the oocyte, so the tail actually falls off. So the mitochondria you inherit from your mother, because those are already present in the oocyte, in the egg cell. Okay. So we're going to talk about embryonic development a little bit. And one of the really important things to realize is that we start out as a single cell. So what that means is that all the cells in your body have the same genetic information, right? So all the cells. So those early cells are sometimes referred to as stem cells. So stem cells are unspecialized, and they can become anything, right? So stem cells are very potent, because you could take stem cells that you might get from the umbilical cord, and you could put them into somebody's brain, and those cells would specialize to become neurons or glial cells, right? They've also found that they can extract stem cells from fat. So people that get liposuction, for example, they're able to liposuction the fat out and find stem cells, and then they can use those stem cells to treat illnesses and disease. So they're very powerful, and it's when we look at, we'll talk about the different types of stem cells, but stem cells can be embryonic, but they can also be adult stem cells. So embryonic stem cells would be in the embryo, but we also have stem cells found throughout the tissues in our bodies that can be induced to become different types of tissues. Okay? So these stem cells differentiate, it's called, and become specialized cells. And these specialized cells have specific genes turned on and others turned off. So this process of differentiation is due to gene regulation. So remember that genes code for proteins. So you can turn genes on. Right, so they can be turned on, and this means that they're producing protein. Or genes can be turned off, right? And that means that they're not producing protein. Right, so you can turn a gene on and off. So, like for example, our pancreas only produces insulin, right? So the gene for insulin is turned on in the pancreas cell, but we also have the exact same gene in our neurons. We have the same gene in our skin cells. We have the same gene in our liver cells, but they're never turned on in those locations. So the pancreas cells are specialized because the insulin gene is turned on, but the pancreas cells they don't ever produce lactase, for example, right? So those genes would be turned off. So this idea of specialization all has to do with gene regulations being turned on and off. Okay. So if we look at this early embryonic development, we start out as a zygote, right? And then we start to divide. The cells will divide, right? And they'll produce two cells. And they're usually kind of found in a membrane. So I'm going to write them like this, okay? So this process of mitosis continues. I don't know why I shouldn't have put 2 lines there, sorry. This process of mitosis continues until we have a solid ball of cells. And they're in a little membrane. Okay. This early stage of development is called a morula. Now, something interesting happens in that this solid ball of cells, so this is solid ball, the solid ball of cells becomes hollow. And so I'm going to kind of draw it in a 3-dimensional plane here. But think of it more like a water balloon. So if it's, this is 2-dimensional. If it was 3-dimensional, it would be a hollow ball of cells, right? So it's fluid filled on the inside. And then it has specialized cells located right here, which are going to become the embryo. So this outer ball, this whole thing is called a blastula. Let me write that again. Blastula. Okay. This is where it leaves the membrane. It has these little cilia on it. And interesting enough, they kind of float around in the uterus before it implants. So it's actually kind of swimming around in the uterus and then it implants. And so this outer part is going to become the placenta. So it's going to actually, I'll put what it's called. It's called a trophoblast. And I'll say it becomes the placenta. Right? And this inner cell mass is going to become the embryo. So now we have like where the cells are located determines gene regulation, right? Because the outer cells are going to become, they're actually going to become the placenta, and the inner cells are going to be the inner cell mass. And then remember we talked about how the embryo that has 3 germ layers, the endoderm, ectoderm, and mesoderm, so that differentiates in here. So this is what implants in the uterus. Okay. So let's talk about what might happen. I think there's some images in your book that show this too. Okay, so this is 2 cells, 4 cells, 8 cells. These would be morula. Right? This is the blastula. The blastula has, you don't need to know this word, but has in the inside of it, it's called the seal, the blastocele. The trophoblast is on the outside, and the inner cell mass is going to become the embryo. Okay, so let's talk about different types of twins. So monozygotic, sometimes abbreviated as MZ. Do you think the monozygotic is identical or fraternal? Mono means one zygote. If it's derived from one zygote, it has to be identical. Okay, so this would be identical twins. So what this means is the early embryo separates into 2. So only one egg was released. Right. Or I guess there could have been more, but only one was released and fertilized. Okay. So they're both from the same egg. So that is opposed to dizygotic. So di means 2, so this would be 2 zygotes. And this is abbreviated as DZ twins. Okay. And so this is where 2 eggs are released. And fertilized. So these 2 individuals, if they're fertilized by the sperm from the same male, right, would be siblings. So they would be fraternal twins. Now there has been instances where females had intercourse with more than one male when they were ovulating, and they had offspring that had different fathers. So you could have dizygotic twins with different fathers if there's 2 different male sperm in the female's reproductive tract. Okay, so this is all kind of pretty normal, but recently they've discovered something else very interesting can happen during early embryonic development, and this is called a chimera. So chimera means that you have 2 different cell types. 2 different cell types. Okay. So this can actually be due to the fusion of 2 embryos. Okay. And Siamese twins, sometimes that happens, but this is where you wouldn't know it by just looking at the person, right? And so this can be, can occur when we have what is referred to as the vanishing twin. I'll put that in. Right. So the twin, they start out 2 embryos and then they produce 1 individual. Right. And so the interesting thing is that if I was a chimera, half of my cells could have the genes of a different person, a sibling, and half of my cells could have the genes of another person. Sometimes you can tell chimeras, like I had a student once who said that when she was born, she had a big mass of tissue on her forehead, and then when they genetically analyzed it, they took it off. It was actually a twin that had just kind of fused onto her during embryonic development. So an example of this recently happened and kind of was in the headlines. right? Where a man was interested in determining whether or not he was the father of his offspring because they had done in vitro fertilization. So they had gone to a fertility clinic and he had provided his sperm, his wife had provided her eggs, and they had produced an offspring. And when they tested the offspring, they found that the offspring was not the father's. And when they did some more subsequent analysis, they discovered that the father was a chimera. So his genes in his sperm were different than the genes that were found in his saliva. So where where one part of his body starts and where one stops is kind of, we don't know, right? But sometimes this can lead to problems with autoimmune diseases. So if you are a chimera and your immune system finds tissue that doesn't appear to be yours, according to the immune system, sometimes it will attack that foreign tissue and cause inflammation. And so chimeras could have a higher incidence of autoimmune disease because they have two different types of cells in their bodies. Now, there's another kind of term for chimera where you actually create an organism like an embryo that has part mouse, part human. That's different than this, right? So chimera mythologically is like one, the head of a horse and the body of a snake, right? But this is just talking about it in terms of two different cell lineages. Okay. Are there any questions about that idea? Chimera?
I mean, would you know?
You wouldn't. So I don't know if I'm a chimera, right? You wouldn't know. So we don't really know how frequently it happens.
In forensics, that would take on a whole different.
That would be a problem, yeah. But you would be able to tell.
I mean, just hypothetically, if that guy raped somebody and they went off the DNA of his sperm.
They would say, it looks like your brother raped somebody. And does he have a brother? And then they would test his brother. So the genetic material is similar.
So he might get off because
He might get off.
they took his DNA from like a cigarette butt.
Yes, exactly. Yeah. that
And
would
they
be-
say, oh, well, it wasn't that.
Right, but it was a close genetic relative.
Oh, so they can't tell.
Right, so the genetic material is going to be the same as siblings. So I could be two individuals, they're not like distantly related. The two individuals in my body would be siblings. My body would be made up of sibling cells.
So they would know that it's still very
Yeah,
close.
so they would go and they say, so then they would do further testing to see, yeah. Okay. Okay. Okay, so we are going to start a new section in class today, which deals with genetics. And so for those of you who don't like memorizing and like problem solving, this will be maybe easier for you. For those who hate problem solving, you're probably going to be like, it's like math class solving, word problems, okay? So, but just kind of approach it as being fun, kind of like a game. So when we talk about Mendelian genetics, we have to realize that Gregor Mendel, was around in the 1850s. And the interesting thing about him is he was actually doing his work. He was a monk and he was doing his genetics work at the same time that Darwin was trying to think about how populations of organisms change, but they never kind of, they didn't know about each other's work. And now we know the significance of, understanding genetics and understanding evolution. Okay, so it's kind of a contemporary with Darwin. So prior to Mendel, we had a theory. And this was what is called the blending theory for inheritance. So the blending theory had a couple of assumptions to it. And the assumptions of this theory, or kind of the predictions, I guess, assumptions, predictions. One is that offspring will always be a blend of the two parents.
Did we just move into chapter 8?
We did. Right? So they'll also always be intermediate. Like, so if you had a red hair and a blonde, they would always be kind of like strawberry blonde, right? So they'd always just be intermediate. And that kind of makes sense from what when we think about ourselves, when we think about the animals that we breed, right? So that kind of makes sense. The second idea is that once blended, traits cannot be separated back out. Now, this poses a big problem if you think about it, because what's going to happen is you're going to lose all variation, right? So if you blend a trait, like if you get red hair and brown hair, and you get like amber or not auburn colored hair, then pretty soon there's not going to be any red hair and brown hair people, because you're always going to have intermediates, right? They're going to blend together and there's no separating them back out, right? So this does not explain variation. So the third thing that the blending theory said is that variation is not important. Right? So these ideas kind of go counterintuitive to what we just talked about, how during meiosis, producing genetically variable offspring are super important for the survival of a population or a species. Okay? So Mendel did some experiments to test this. And he used pea plants so that he could artificially take the sperm from one flower, which is called the pollen, and pollinate the ovaries in another flower and get genetic crosses between pea plants. And so one characteristic that he looked at, or trait, was plant height. Okay, and the plant height in pea plants that he was looking at was tall and short. So some of his pea plants were tall and some of his pea plants were short. And so he did what is called a cross, where he took pollen from one plant and he pollinated the seeds or the eggs from another plant, right? So we did a genetic cross, okay? What he found when he did this cross is that all of the offspring were tall. And you're like, why aren't they medium height, right? He was smart in that he picked traits that were not variable. So there was not a continuous variation in height like there is in humans. You either had tall plants or you had short plants. And so that's kind of the way, it was a very simple model so that he could make some discoveries about the way that traits are inherited that you wouldn't be able to do if there was lots of variation in these traits, okay? So then he took these offspring from that cross, and he crossed them again. So this means that we're mating them. This little X means that I'm mating them, right? And then what he got was tall and short offspring. Okay. So this defies the second one, right? So this was the idea that once the trait has been blended, it cannot be separated back out. But now we see that it is separated back out, right? And so variation is maintained in this process of passing traits from one generation to the next. We maintain variation, right? We all don't become more similar the more generations there are, okay? So if we go back to this is what is referred to as the parental generation. Okay. And we also say that these parents are true breeding. What that means is that if I planted seeds from a population that is only tall, I will always get tall plants. If I do it for short, I will always get short plants, right? So the offspring that are produced, this is called filial 1, so it's F1, just knew F1, that's filial means offspring. F1 generation, right? These are hybrids, right? So if you're a gardener, you know that sometimes you get hybrids when you don't want them. So for example, I wanted these really beautiful dark maroon hollyhocks, right? And I bought them and I planted them and they were beautiful the first year, but then they produced seeds the next year, and what I got were like maroon and yellow and kind of an icky orange, right? And so I got all this variation, and the reason was because the plants that I had planted were hybrids, right? And so they produced more variation in the next generation. So sometimes you see on seed packets, you see this is a hybrid, and you know if you want that trait, you have to buy the hybrids every year. Because if they reseed themselves, you're not going to necessarily get that trait again.
Is that the same concept? Because we used to breed Labradors,
Mm-hmm.
and they say hypothetically that breed Labradors you never can guarantee because you can't go to the true. I mean, you don't know where the true breeding starts with Labrador. You can have two really good AKC papered black labs.
Yes.
And they can throw a chocolate away.
Right. Yes. And that actually means that technically means that your dogs were probably a little healthier. If they have a little bit of hybrid, like we need to. So hybrids tend to be healthier, plants and animals. So hybrids are not inbred. So you can think of it as like the difference between a mutt and a purebred. Purebreds tend to have less genetic variation, and so they tend to have a lot more health problems. So it's actually better to have more genetic variation in your dogs than less.
Because they said, I mean, they always said, as far as being papered, because at first we were like, how did we get two black ones? How did we get a white and chocolate?
Yeah, I mean, we'll talk about inbreeding, but when we get to pedigree.
But they were like still right pedigree, right?
Right. Yeah. Okay. This then is the F2 generation. Okay. So sometimes when you hear about genetic crosses, they call this cross right here a monohybrid cross. Mono means that it is a single gene. So we're looking at the gene for height, right? And it's said to be a monohybrid. It's only one gene. We're only looking at one trait. And we're crossing them to see what the F2 generation is.
So that is for the F1 generation?
When you cross it, a hybrid tall with a hybrid tall, that is called a monohybrid cross.
Oh, when they're the same,
Yes.
process, the same.
Okay. Yep. Okay. Versus what we'll talk about dihybrids later. Okay. Okay, so now we need to talk about some terminology. Okay, so we can talk about allele. Okay. So allele is an alternate form of a gene. Sometimes in the most simplest examples, like with Mendel, he had a dominant versus recessive. So the dominant allele is a capital letter, and the recessive allele is lowercase. So for example, we could have the gene T for height, right? This is my gene. The dominant appears to be big T, right? Because that codes for tall. And then we would use little t, which produces short. So there's one gene for height, One form of that gene produces tall plants. One form of that gene produces short plants. And so the big T and the little t are said to be alleles, dominant and recessive. I'll explain that in a minute. Okay, so we could talk about the genotype. So the genotype is the combination of alleles. That an organism has, right? So for example, we could have homozygous recessive. So just remember, homo means same, and so I'm saying that it is recessive, so I would have 2 copies of the little allele. Notice since I am diploid, plants are diploid, so they have to have 2 copies of every gene, right? And those genes would be on separate chromosomes, right? So the chromosomes would look like this. You have a little t and a little t, right? Because we have 2 copies of every chromosome, hence 2 copies of every gene. We could also have homozygous dominant, and that would be big T, big T, big T, big T, big T. And then if they are one of each, does anybody know what that's called? Heterozygous. Hetero means other, like heterosexual, male and a female, so you have one of each. You have a big T and a little t. Okay, so that is heterozygous. So those are the genotypes. Okay. The phenotype, on the other hand, is the outward expression of the combination of alleles. All right, so that's the phenotype. So in the example that I gave, we talked about tall versus short. But it doesn't necessarily have to be something that I can look at and see on the outside of your body. So it could be, oh, do you produce lactase? Do you have that gene that allows you to produce lactase to digest milk? Or do you not, right? That's still outward expression because it's inside your body, but it is expressing itself outside of the genome, right? Okay, so let's go back to that image or the initial cross that we did and draw in the genotypes. So you'll notice that I only said phenotypes. So this is phenotypes, tall versus short, tall, tall, and short, okay? So in a different color, you could do this on a separate page if you wanted to. If it is true breeding for tall, what do you think its genotype is? True breeding for tall, what do you think the genotype would be?
Big T, big T.
Yep. If it is true breeding for short, oops, you're right, little t, okay. Now, think about how the chromosomes behave during meiosis, and also remember that Mendel had no idea what the genetic material was. He didn't ever see a chromosome, he's just putting stuff in and seeing what comes out, right? And so those plants would produce only gametes with the big T. These plants would only produce gametes with the little t. So these hybrids have to be big T, little t.
I have a question.
Yep,
To find out, do you have to use the Punnett squares?
we will use a Punnett square, but we're not there yet. We're not quite there yet. Okay, so these individuals that are tall, notice that they could either be big T, or big T, little t. So these guys, we cannot tell just by looking at them. They could either be homozygous dominant or heterozygous. I can't tell. These guys would have to be little t, little t. So that would be my genotypes in this particular example. But obviously, Mendel didn't go that far because he didn't know what he called these factors that were separated out and came back together. Okay, so applying this to humans, we do not, do experiments like plants. We don't artificially inseminate because it would take too long because we don't produce that many offspring, right? So what we do instead is we do what is called the pedigree analysis for humans. And so when we look at a pedigree, a square is a male. A circle is a female. We can say that a male and a female mate by putting a line between them. Does not necessarily mean marriage, but it means that they are mating, right? And then they can produce offspring. Okay. So this is a male and a female that produced a male or a son and a daughter, right? And then that son and daughter could also, let's say this daughter also has a mate and also produces an offspring. Okay, so what this tells me is that this is generations, right? So this is generation 1. So in generation 1, I number it 1, 2. Okay, this is generation 2, Roman numeral. Sorry, this should be 1, Roman numeral. This is 2. This is 3. Okay, in generation 2, sorry, I have 1, 2, 3, and generation 3, I just have 1. Okay, so I can point to individuals on my pedigree by saying individual 3, 1. Right, and I know that I am talking about that person. Now, when we do pedigrees, we're interested in particular traits, and they could be diseases, or they could be like physical characteristics. So what I would do is I would color in the individuals with the trait of interest. Okay. So this is an individual with the trait of interest. Okay, so it could be a disease, or it could be like the ability to taste a chemical. We'll do that in lab today. Okay, it could be other things. It could be color blindness or something like that. Okay, so that is what a pedigree does. So if we look at how pedigrees can tell us information, we can look at the patterns of inheritance of particular traits. Okay, so we are going to first look at autosomal patterns. And this is the gene has to be on a chromosome other than the sex chromosome. X or Y. And so those are called autosomal chromosomes. Okay. Maybe, I don't know what self-body, I don't know what autosomal means, self-body. Okay. This is versus sex-linked. And this is where the gene is on the X or Y chromosome. Most sex-linked traits are actually, we'll talk about this not today, but most of them are X because X is so much bigger than the Y. Although being a male and a female, we're going to look at in lab today, that actually depends upon a gene that is on the Y chromosome. So this is one way we can describe patterns of inheritance as autosomal or sex-linked. We can also describe them as dominant versus recessive. Yes, that is. That's a pattern that you sometimes see. Yeah, it skips generations. Yeah. Okay. Okay, so let's look at an example of putting this together. Autosomal recessive trait. Okay, so it is caused by a gene on not the X or Y chromosome, and is caused by the trait that we're interested in is caused by a recessive gene. Okay, so if we look at an example of this, we can look at a trait like albinism. Now, there's different forms of albinism, but the most common form is autosomal recessive. Okay. So if I drew my pedigree, and actually let's do one more generation, sorry, one more generation, so we can show an uncle or an aunt. There we go. Okay, so this is the son of these two individuals, right? Actually, this needs to go to the daughter, sorry. Okay, so this needs to go to this one. Okay, that looks better. Okay, so that's the son of those two individuals. This is his aunt, right? These are the individuals that have albinism, right? It's an autosomal recessive trait. So what we know is that it's caused by the recessive allele. So we could give this gene any letter we wanted to, but we could just give it A for albinism, right? So this is the gene A, and so I always I do this on all my genetics problems. I put a little legend in there because this helps me remember. So A, big A, means that is normal because it's a recessive trait. So the dominant allele is going to code for normal. The recessive allele is going to code for albinism. Okay. So what must be this would be generation 3, number 1. What is his genotype? Big A, big A? No. Little a, little a. Okay. Homozygous recessive, that's a little a, little a. Let's write that over again. Okay. Now, we have to kind of work backwards. We know she must also be little a, little a. Okay. Does that make sense to everybody? Okay. So what must the parents be if they have an offspring that is little a, little a?
Big A, little a, big A, little a.
Yes. And so we say that in this example of a recessive trait, these individuals are carriers. Right? They carry the trait, they don't have it, but they carry it to the next generation. Okay? What about these two individuals? Can you give me any information about those two individuals in generation 1? The grandparents?
Big T, big T, little T, little T.
Well, A, yeah. Perfect. So they are also carriers because they had a child, right? The aunt that also had albinism. So albinism runs in this family, in this pattern. This individual, we have to do a Punnett square. Okay. So in a Punnett square, you do a little cross here. So these are the parents. Okay. This individual, 50% of its gametes are going to have a big A. 50% of the gametes are going to have a little a. And I don't need to worry about egg and sperm, right? Because this is autosomal. It doesn't matter. Sex chromosomes do not matter. Okay. So these would be the gametes of the first individual, right? So 50-50. just because of the way the chromosomes separate during meiosis. And here would be big A, and here would be little a. And then I put them together in a square. So those are the gametes. I'm just simply doing multiplying probabilities here. So 50% times 50% is actually 0.25. So I put big A right here. Okay, that's 1 out of 4 would be big A, big A. Big A, little a. Big A, little a. Notice that I didn't put little a, big A, just because it's harder to read. So I always put the dominant allele first, and then little a, little a. Okay. Now, does that daughter have albinism? No. So I can actually get rid of this probability, right? So what is the probability that she is not a carrier? 1. She's not a carrier. 1 out of 3. Okay, because I got rid of this possibility.
Why did you get rid of that?
Because does not have albinism, so I know that this is not a
But aren't
possibility.
the original odds still, she's still got the odds?
No.
But we know that that's not it, so we've then narrowed it down to this.
So you narrow it down. So that she's not a carrier is 1. This is not a carrier out of 3 possibilities. So 1 out of, so third. Right? That she is a carrier would be 2 out of 3. Okay. So she's actually more likely to be a carrier. So you can put 2/3 carrier probability, right? And 1 out of 3 probability for normal. Put normal. Oops, my pen won't work now. It's because I have oh, just write normal there. Okay, 1/3 normal. And you can put those into decimals, so it could be 0.666, right? 0.333. Okay, so those guys have another child, okay? So let's say that they had another offspring. So I could put that one over here. So if they're worried about, they say, we want another offspring, they go to a genetics counselor and they say, what is the probability if we have another offspring that that offspring would have albinism? What would it be?
2 out of 4.
No. 1
out of 2.
1 out of 4. Right? So it'd be a 25% chance, 1 out of 4 chance that they would have, if they had another child, that that child would have albinism. Doesn't matter that they already had one, a child with albinism, because it's independent events. It's odds are the same. It's like flipping a coin. Odds would be the same.
Why wouldn't the same odds, why wouldn't the same odds appear for the other sibling?
We have no information about the sibling, so we can't rule out one example. Every one of those offspring that were born had a 25% chance of having albinism. This one definitely has it. This one definitely doesn't. This one's complete unknown.
Got it.
Okay. So notice that this skipped a generation. That is common for autosomal recessive. So recessive traits, okay, tend to skip generations. So if we look at another example, okay, so this is a disease called Huntington's disease, which is a neurological disorder that kind of manifests itself in people's 40s and 50s, and they start to just have this neurological motor, it just degenerates and they die of it. So it is lethal. So when we see a lethal trait, or if somebody dies of the disease or the trait of interest, we put a cross through that, okay? So that means that person died of Huntington's. Okay. This is autosomal dominant. So this is not recessive. There are no carriers. It does not skip generations. Okay. So now I use my legend again because this is really important. So this is my gene. My gene is is H, right? So my gene is H. So in this case, big H is the disease because it's dominant. This disease is dominant, okay? In this case, little h is normal, okay? So now I go through and I say, oh, what's really I can tell when a person doesn't have the disease, then they have to be big H, or excuse me, little h, right? They do not have the disease. So now it's the ones that are not colored in that are autosomal or homozygous recessive. Okay. How about this individual? What must they have been? Could they be big H, big H? They have a child that doesn't have it? Okay. How about this one? Could it be big H, big H? No, right? Because it has a parent that doesn't have it. Okay. So what was the probability that if they have another child, or if they did have another child, since this is a late onset, if they had another child, what was the probability that that child would have the disease? So we could do our Punnett square. Okay. And if you're not used to it, you might want to do it a couple of times. Okay. Setting it up real simple, real basic. Those are the gametes. They come together. And remember, I always put the big H first. So what was the probability that if they had another child, that that child would have Huntington's disease? 50%. And so they can do, the question is, people who have this running in their family, you could go get genetically tested, and if you have the big H, you are going to die. If you don't die sooner, you will die by the time you're 55, right? And so the question is, would you, knowing that this runs in your family, would you be tested? And is it kind of important for you to be tested if you were going to have children, right? So like, for example, if this person was going to have children, he could pass that lethal allele onto his offspring. However, you might be able to screen and make sure that only sperm that have the normal allele are the ones that fertilize the egg. So you could do some probably expensive genetic screening to try to ensure that if you did have Huntington's disease, that you did not pass it on to your offspring. Okay.
I have a question.
Yeah.
Traits manifest themselves differently, like if it's the same trait and it manifests itself differently because
Yes. Yeah. So there are different, like for some of the diseases, there's more than one allele. So I'm just saying there's 2 alleles, but like for cystic fibrosis, there's like 7 different alleles. And so it depends upon what mutant allele you get that determines whether you have severe cystic fibrosis or if you have mild cystic fibrosis. So there's, in some cases, there's multiple alleles. And it can also, there's environmental, so the genes interact with the environment.
are we gonna learn more about that word?
Yeah, a little bit. Yeah.
I don't know if that's, I'm just curious.
Okay.
My son, we just found out something, and I have, so kidneys think that it's genetic from him, but because of, well, the things that occurred. So the one is that, yeah, it's been a different, like, it's been different from person to person. Like what my son has is not what they have, but it is all
Related.
similar, it's a kidney, something related to the kidney.
Yeah.
But his is totally different than some that, you know, we've heard of, different kidney things that if you have a genetic kind of problem with, you know, kidney related, it manifests itself
Yes.
differently. So I,
Yeah. So we'll talk about it. It's called genetic heterogeneity. Yeah. So maybe we'll get to that a little bit. Probably.
and we're gonna participate in a study at OHSU with him to help find out if it is genetically related, which